This lecture is continuation of my previous lecture on eukaryotic gene expression 1. That was a basic one. Here I will discuss some more aspects of eukaryotic gene expression. In eukaryotes, expression of any gene begins when some activator binds to the enhancer element present in the DNA. And this activator has a DNA binding domain and a protein binding domain. It activates the binding of mediator, modification and remodeling enzymes and other transcription factors. When they come into place, they attract the general transcription factors of RNA polymerase 2 like Tata binding protein, TBP, and then other transcription factors. These are general transcription factors. They also bring in transcription factors, as well as this TF2F contains RNA polymerase 2. And finally, when whole thing is assembled, transcription factor 2H performs a helicase activity as well as for phosphorylation of the CTD, and transcription begins. Now, this part I have discussed pretty much in detail in my last lecture, but here I will discuss what makes this activator proteins to bind to the enhancer elements. What are those signals which initiate this whole cascade? There are inter and intracellular signals that activate this cascade of gene expression in eukaryotes, and we can divide it into two major components. One are like steroid-like molecules, steroids, retinoid, and thyroid hormones. They can cross the plasma membrane by simple diffusion because of their nature, which is hydrophobic in nature and similar to the plasma membrane. And they reach the nucleus and bind to specific receptor proteins over there. And hormone receptor complex binds to the specific DNA sequence, which is called as hormone receptor elements. So they bind to this specific receptor protein, which ultimately binds to the specific DNA sequence. So here I give you some examples. These are the hormone response elements which are bound by steroid type hormone receptors. These are names of the few receptors which are steroid type, and these are the consensus sequences of the DNA where they bind. This is general mechanism by which steroid and thyroid hormones, retinoids, and vitamin D regulate the gene expression in eukaryotes. There are serum binding proteins which bind these hormones from where they are secreted, and they are carried in the blood to the destination place where this hormone is released. It crosses the membrane, the plasma membrane, and then the nuclear membrane. Inside the nucleus, this hormone, it binds to its receptor, which is specific for this hormone. When this receptor is bound by this hormone, there are certain conformational changes that take place in this receptor molecule. And now this receptor molecule, along with the hormone, can bind to the hormone response element, which is present on the DNA molecule. As I mentioned in the previous slide, these are certain sequences which are specific for specific type of hormone and their receptors. And when they bind here, the same cascade is initiated where mediator and chromatin remodeling enzymes and other proteins and transcription factors come into place. Finally, RNA polymerase 2 binds and starts the transcription, and followed by translation, synthesis of the protein. These are the features of hormone receptors which bind to the hormone. These are protein in nature. This is the N-terminal site, and this is the carboxy terminal end of the protein, and there is a transcription activation domain with a variable sequence. Then there is a DNA binding domain. It is 66 to 68 residues, and it is highly conserved. Then there is a hormone binding domain, which is variable sequence and length. And this hormone receptor, it has a highly conserved DNA binding domain with 2 zinc fingers. These are the 2 zinc fingers in which zinc is present inside. So these are the transcription factors, and this zinc is coordinated with cysteines. These are few of the important properties of the ligand binding region of the receptor proteins. This ligand binding region is usually present at the carboxy terminus, and it is quite specific to particular receptors. As I mentioned earlier as well, this glucocorticoid receptor only 30% similar to estrogen receptor, and only 17% to thyroid hormone receptors. Hence, due to this difference and specificity, specific types of receptors can only bind to the specific genes for which alteration in expression is desired. And size of the ligand binding region also varies from 25 amino acid residues in, for example, vitamin D receptor, 603 amino acid residues in mineralocorticoid receptor. So you can see how much variation in the ligand binding region that takes place. And there is an unusual coactivator, which is a steroid receptor RNA activator. It is 700 nucleotide RNA, which acts as a part of a ribonucleoprotein. Meaning of this is that RNA can also act as a receptor along with the protein molecule. Now this is an example of a hormone which cannot cross the membrane and is hydrophilic in nature, like insulin. So regulation of gene expression in insulin is brought about by mitogen activated protein kinase cascade, which is called as MAP kinase cascade. And this protein, insulin, it binds to some protein which are present in the plasma membrane. These are transmembrane proteins, and this insulin binds to the external side of these proteins and bring conformational change inside the protein. That, for example, in this case, this protein is phosphorylated. It activates, this phosphorylation activates further transcription factors, as this transcription factor is phosphorylated, and so on. This whole process, a number of different proteins are phosphorylated and dephosphorylated, and finally one of these proteins, ERK in this case, when it is phosphorylated, it can cross the nuclear membrane. Once inside the nuclear membrane, inside the nucleus, they perform the similar function as I discussed earlier, that they initiate the transcription factors to bind at this region, followed by expression of the RNA polymerase 2, and this transcript, it comes out of the nucleus into the cytoplasm for protein synthesis. Carbohydrate response element binding protein is a transcription factor, and as long as this is fully phosphorylated, it cannot cross the membrane, the nucleus membrane. So because it cannot cross the nuclear membrane, therefore the transcription attached to this transcription factors cannot take place. So how it is activated? It is activated with the help of glucose, which binds to a specific transport proteins, glucose transporter. This is one of those. So once glucose is inside the cell, it goes certain transformations, and finally it can uh, dephosphorylate this carbohydrate response element binding protein to some extent so that it can cross the nuclear membrane. Once inside the nucleus, the other phosphate is also released, and now this carbohydrate response element binding protein interacts with other proteins, uh, meaning this is responsible for initiating the gene expression related to this transcription factor. So this carbohydrate response element binding protein, it is bound to the DNA at specific place after it is binding to the receptor protein, and once it is bound there, then there is a cascade of reaction takes place by binding the transcription factors, mediator, and finally RNA polymerase 2, which performs the function of transcription. Here is another example in which insulin comes outside the cell membrane and it binds to its receptor. In this case, the transcription factor which is activated is called as FOXO1. And this FOXO1, this is quite reverse of the previous example, because in this case, this FOXO1, when it is phosphorylated, it is degraded in the cytosol. And it can only cross the membrane when it is dephosphorylated. So as opposed to carbohydrate response binding element, FOXO1 crosses the nuclear membrane when there is no phosphate attached to it, and the other function remains the same, the binding of the transcription factors, mediators, and finally RNA polymerase to do the transcription. This figure shows complexity of the transcription factors in case of eukaryotic cells. This is just an example of phosphoenol pyruvate carboxykinase promoter region, and you can see how many different protein factors bind at this place, including this FOXO1 and many others, that eventually attract mediator and other transcription factors, and finally RNA polymerase 2 to perform the function of transcription. So this shows how complicated the expression of gene in case of eukaryotic cells as compared to the prokaryotic cells. So you have to look for so many factors which are bound to this promoter region for transcription to occur. Ethylene also can signal transcription in case of plants. For example, ethylene comes on the outer side of the membrane and it brings about certain changes, conformational changes in the protein, which in turn binds to the hormone response element in the DNA molecule, and finally there is transcription that takes place. I'm not going into much details of what are these proteins, but overall message is same. This is an example of another signal molecule such as erythropoietin, which has affinity for a membrane protein on one end, and there are certain changes that are brought about on the other end of this protein. And this is called as JAK-STAT transduction mechanism for this erythropoietin receptor, which is this one. So which it attracts the erythropoietin, and then there is a certain change, phosphorylation of the JAK protein that takes place. Then there is similar MAP kinase cascade that is brought about, and different proteins, they follow this path, and finally It affects gene expression inside the nucleus cell in case of eukaryotes. There is a long list of transcription factors with significant roles in plant stress tolerance. So this is from one of the papers. I will share the link. But you can find different examples not only from plants but also from animals as well. Now coming to the regulatory proteins which actually are performing this function of eukaryotic gene expression, controlling the eukaryotic gene expression. The properties of these regulatory proteins are they are 10 raised to power 4 to 10 raised to power 6, meaning 10,000 to 1 million times higher affinity for their specific sequences on the DNA molecule. There are discrete DNA binding domains which are present in these regulatory proteins, and they include one or more of a relatively small group of recognizable and characteristic structural motifs. Amino acid side chains have hydrogen bonding to DNA bases with asparagine, glutamine, glutamic acid, lysine, and arginine residues. However, there is no simple amino acid base code that we can say for sure that this is the code for amino acid and base to bind for such kind of regulatory proteins at the time of gene regulation. Here are the two examples of specific amino acid residue interaction with the base pair, like this glutamine or asparagine. It can have such kind of hydrogen bonding interaction with these nucleotides and or nitrogenous bases. So here you can see with cytosine and guanine, there is an interaction of these hydrogens, and this is an hydrogen bonding with the arginine. However, once again, I want to mention is that there is no specific code that we have by which we can determine which protein can bind to which DNA molecule for this purpose of expression and regulation. DNA binding domains of these regulatory proteins tend to be small, like 60 to 98 amino acid residues. And structural motifs within these domains are still smaller than this. The DNA binding sites for regulatory proteins are often inverted repeats of a short DNA sequence, and which are called as palindromic sequences, at which multiple subunits of regulatory protein bind cooperatively. Cooperatively meaning if one protein subunit is bound, the binding of the other protein subunit is enhanced. So there is a cooperative binding. Lac repressor of E. coli, however, functions as a tetramer, as we have discussed already in one of my lectures. There are several types of DNA binding motifs which perform their function during gene expression. But here I give you the example of the most common one. The first one is helix-turn helix. It contains 20 amino acids in two short alpha helical segments. So both have the helical structure, each 7 to 9 amino acid residues long, separated by a beta turn. Zinc finger is another transcription factor which is about 30 amino acid residues long, and this is held together by zinc ion, hence its name, and it is coordinated to 4 of the residues, either 4 cysteines or 2 cysteines and 2 histidines. Zinc does not interact with DNA, but it stabilizes this transcription factor. Multiple zinc fingers are found in DNA binding proteins. Another example is the homeodomain. This is a type of DNA domain which is a transcriptional regulator. It is about 60 amino acid long and it is highly conserved in eukaryotes. DNA binding segment of this domain is related to helix turn helix motif, and this is the structure uh, for this homeodomain. For protein-protein interaction, two important structural motifs uh, mediating the protein-protein interactions, they are called as leucine zipper and basic helix loop helix. These two are for the protein-protein interaction only. No DNA is involved in this case. Leucine zipper is another transcription factor which is involved in protein-protein interaction, and this is an amphipathic alpha helix, and it has a series of hydrophobic amino acid residues which are concentrated on one side. The alpha helices have leucine residues after every 7th position, and it forms a straight line along the hydrophobic surfaces, as you can see here in this figure. A zip-like structure is there. Regulatory proteins with leucine zippers often have separate DNA binding domains with high concentration of basic amino acids, lysine or arginine. Leucine zippers are found in many eukaryotic and prokaryotic proteins. These are few of the examples of leucine zipper transcription factor. This is the connector side, and here is the DNA binding region. So this region binds to the DNA, and this is for the protein-protein interaction. And these examples are taken from mammalian cells, JUNFOX, and these are different transcription factors that contain this leucine zipper. Similarly, in yeast, it also exists, and this is the consensus sequence. And as you can see, there is leucine at every 7th nucleotide that makes this zip-like structure. Basic helix loop helix is another structure, and this motif is found in eukaryotic regulatory proteins. It is conserved region of about 50 amino acid residues, which is important in both DNA binding and protein dimerization. Two short amphipathic alpha helices, they are linked by loop of a variable length in this basic helix loop helix structure. There are certain additional domains for protein-protein interaction. At least 3 types of additional domains which are characterized primarily in eukaryotic cells are glutamine-rich, proline-rich, and acidic domain. I have discussed their functions in my previous lecture on eukaryotic gene expression 1. Here you can see some of the examples of the activator proteins. They are bound to the US, upstream activator sequence. So if we take the example of this GAL4P, which is for galactose metabolism, it contains a zinc finger-like structure in its DNA binding domain. And it binds to this upstream activator sequence, which is a palindromic sequence of about 17 base pair. And it has acidic domain towards a protein-protein interaction. So I have given is more detail in my lecture on eukaryotic gene expression part 1. You can see from there the detailed mechanism of this GAL4P. SP1 is a transcription activator for a large number of genes in eukaryotic cells. DNA binding site is GC box. It contains these G's and C. It is present near Tata box. Its binding domain contains 3 zinc fingers of this SP1, and 2 other domains function in activation. 25% of their residues are glutamine. Thus, it is a glutamine-rich domain that functions as a protein-protein interaction in this case. Similarly, another example of CTF1. This is a C-CAT binding transcription factor 1. It belongs to family of transcription activators. Bind a sequence which is CCAT, C-CAT sequence. Its consensus sequence is this one. And DNA binding domain contains many basic amino acid residues. And its activation domain for protein-protein interaction is proline rich. More than 20% of the amino acid residues contain proline. Now coming to the translational control of genes. So far we have been discussing control at the transcription level, but in certain cases in eukaryotic cells, translational control also exists. Translational initiation factors are subject to phosphorylation by protein kinases. And these phosphorylated forms are often less active and cause a general depression of the translation in the cell. It means this is the first control, that if the translational initiation factors are phosphorylated, translation is repressed. Similarly, some proteins bind directly to messenger RNA and they act as translational repressors, many of them binding at specific sites in the 3' untranslated region. So positioned, these proteins interact with translation initiation factors bound to mRNA or with the 40S ribosomal subunit to prevent translation initiation. As you can see from this figure, that there is a binding of this poly A tail, and then there is a 5' cap that binds at this place. In my lecture on translation, I have discussed its details. So here is the 3' UTR where these translational repressors can bind, and they can perform this function of translational repression if needed in certain cases. Binding proteins present in eukaryotes from yeast to mammals disrupt the interaction between these eukaryotic initiation factors, and these mammalian versions are known as this is the name of the mammalian versions. When cell growth is slow, these proteins limit translation by binding to the site on eukaryotic initiation factor 4E that normally interacts with F4G. So when cell growth is slow or increases in response to growth factor or other stimuli, the binding proteins are inactivated by protein kinase dependent phosphorylation. So it means the translation, it follows the level of the cell growth, and it depends on the cell growth whether it is slow or it is faster. So these translational control act accordingly with reference to the cell growth. Then there is RNA-mediated regulation of gene expression, that also this occurs at the translational repression. This is how this translation is repressed by RNA mediated, as I mentioned in the last slide, in the last point in the last slide. So here is with the help of this primary microRNA. They are produced by RNA polymerase 2 in eukaryotic cells, and they also have this poly A tail. And they are converted into pre-microRNA with the help of this protein, and in the form of pre-microRNA, they are exported out of the nucleus and they come into the cytoplasm. In the cytoplasm, there is a dicer acts over there, and dicing basically of this microRNA takes place. So it means this is a single RNA molecule which is having intra-chain hydrogen bonding, but with the dicing, there is a cut at this place, and they become double-stranded. Now from single-stranded to double-stranded with interchain hydrogen bonding. and this is called as microRNA duplex. And then with the help of a helicase, there is unwinding takes place, so both of these strands are separated. Now these strands, one of these strands has an can interact with the messenger RNA to stop translation, but this is brought about by this RISC, RNA-induced silencing complex. There are certain proteins which actually capture these microRNA, and then there are two possibilities here for the microRNA. One is that there is a partial complementarity of the messenger RNA to this microRNA bound by the RISC. So if this partial complementary, then there is a translational repression takes place, meaning there is no translation from that particular messenger RNA. But in case of near perfect complementarity, what happens? Certain enzymes, they act on it, and there is a cleavage that takes place altogether. So in this case, there is no translation as well. So this translation can be repressed in both cases, but a slightly different mechanism depending on the level of complementarity between the microRNA and the messenger RNA, which is going to be expressed inside the cell. Similar type of mechanism also done in vitro. You might know about the RNAi technology, but if we do that by ourselves, that is an RNAi. But here is the example of microRNA, how they play their role. So we call in this case in vivo, we call it as microRNA. Here's a comparison of the two. Here, this is a precursor primary microRNA, and the mechanism I have discussed, and this is called a small temporal RNA, followed by the same mechanism of partial or near perfect complementarity. But in case of in vitro, there is a duplex RNA, we call it as a duplex RNA. Then there is a dicing, and it is called as small interfering siRNA, and which also follows the same pathway later on. So that can be used in different experiments. Now comes another type of gene expression, catabolite repression, and I will give the examples of fungal gene expression here, especially with reference to this trichoderma cellulases, which are most well understood mechanism in terms of this catabolite repression in case of eukaryotes. We already discussed catabolite repression in case of prokaryotes in lac operon system. Here, although this paper is quite older one, but it gives us a fundamentals of this molecular biology of this very important industrial fungus, and a good example of catabolite repression in case of eukaryotes. Cellulases are the enzymes which hydrolyze cellulosic material, and they are found in fungi and in bacteria as well, but we will discuss their gene expression in case of fungi. There are three types of cellulases which are produced by at least three different types of genes. There may be more isomers of these. One very good example of cellulase production, which is of industrial importance, is the Trichoderma ricci. Its older name now is called as Hypochondria jacorina. This is the most studied fungus for cellulase production and being used in industry. As I mentioned, that glucose, when it is present inside the cell, there is no need of this cellulase gene expression, because what is the function of these cellulases? They go out of the cell into the medium. They convert the cellulosic material into sugars, glucose, which comes inside the cell for metabolism. But in this case, if glucose is already present inside the fungal cell, there is no need of gene expression. So glucose represses the cellulase production. An inducer of the cellulase gene is the molecule which is called as sophorose, beta 1,2 glycosidic bond. It is a natural inducer of cellulase genes, we can say, and it is found by transglycosylation of cellobiose. Cellobiose is composed of beta 1,4 linkage between two glucose molecules and the basic subunit of the cellulose. So first we discuss one of the promoters of cellulase genes. This is cellobiohydrolase. As I mentioned, there are three types of cellulases. So cellobiohydrolase 1 promoter. When there is glucose is in enough quantity inside the cell, the cellulase gene is repressed, and this CBH1 is repressed with the help of a repressor protein, which is CRE1. It is very similar to CREA of Aspergillus nidulans and MIG1 of S. cerevisiae. It means that such type of repression is found in other fungi like this Aspergillus and also in yeast, Saccharomyces cerevisiae. This CRE1 is a phosphoprotein, and when there is more concentration of glucose inside the fungal cells, this glucose phosphorylates this CRE1 at serine 241, which binds to the promoter region. So this protein can only bind to the promoter region when it is phosphorylated at serine 241, and this is brought about with the help of glucose by some mechanism. So two CRE1 binding sites are there on CBOH1 promoter, and these sites are present upstream, one at -700, another one is -1000. What it means is that these two CRE1 binding sites are there where this CRE1 is bound when it is phosphorylated, meaning when glucose concentration is more in the cell, and it represses this CBH1 promoter. Now, how does this activation of CBH1 takes place? There are two activators. One is called as ACE1, activator of cellulase gene expression. This is a DNA binding protein having three zinc finger motifs of this nature, two cysteines, two histidines. At least 8 binding sites of ACE1 are present in CBH1 promoter, and they recognize this and this sequences. Then there is an ACE2, activator of cellulase gene 2. It also binds to a certain region, which is, this is the sequence, and it is at -779, and this is also a zinc finger protein. So these two proteins, they act as activator of the CBH1. CBH2 cellobiohydrolase 2 is another gene which is present in this fungus, and it has this CAE, CBH2 activating element. This is a nucleotide sequence. This is the sequence that binds to the protein complex. And at least one copy of CCAT in the template strand or GTAT on coding strand is needed adjacent to this CAE. You might remember that in my lecture earlier, I discussed about CCAT, which is a sequence present in the DNA molecule where activator proteins can bind, and this is the same CCAT site which is present in template strand, or its complementary strand in the coding strand, and it is near this CBH2 activating element, which is also a sequence over there. So different proteins bind to CAE, and CCAT motif basically it binds to this heme activated protein. HAP235 is the name of this protein, and then HAP4 binds directly to this trimer. So there is an interaction of these different proteins to this CCAT, and which in turn has an interaction with the CAE for activation of this CBH2 cellobiohydrolase 2. Apart from cellulases, another important gene is xylanase. There are different xylanases which are also present in fungi. Xylanases degrade hemicellulosic content. So you can imagine, since this cellulose and hemicellulose both are present simultaneously, so there need to be activation of xylanases as well as cellulases for complete degradation. And but this hemicellulose basic ingredient is xylan, which is a 5-carbon sugar, whereas in case of cellulose, it is of course a glucose with 6-carbon sugar. This is the major difference. But these hemicellulose also has long chains of this xylose. And there are two promoters, XY1 and XYN1 and XYN2 promoters for xylanase gene expression. And there is XLNR is a transcriptional repressor of Aspergillus niger xylanolytic system. So Aspergillus niger is also a fungus which produces these xylanases. It is well known producer of xylanases. Some of the fungi, they produce more xylanases, other produce more cellulases. It is zinc binuclear cluster protein. It controls the transcription of many xylan degrading genes, also controls EGL-A and EGL-B genes. So you can imagine from here that this endoglucanase A and endoglucanase B are the two cellulases genes, just like I just mentioned cellobiohydrolase. Another one is endoglucanase, so it is different isomers of endoglucanase. So these two cellulase genes are also controlled by the same transcription factors which are controlling the xylan degrading genes. So both they act simultaneously. In this figure, you can see there is a repression or induction of the genes. So under high glucose concentration, as I mentioned earlier, this CRE1 is phosphorylated. And although these proteins are bound, then there is XYR1 is also there, but this system is at the basal level of expression, at very low level of expression. But when this xylan or xylose come in place, this CRE1 is dephosphorylated, it is not bound here, and there is some signal which also activates this XYR1, and these proteins are already there. So as I mentioned previously, that CRE1 acts for repressor in case of cellobiohydrolase, but it is also acting simultaneously for these xylans and this xylose. Xylan is the long chain of this hemicellulose, and this xylose is the 5-carbon sugar, which is the basic sugar which is present in case of hemicellulose. So this is how this activation and repression goes on. And for XYN2, again, you can see in non-induced form, when there is enough glucose is there, and experimentally people have done in the presence of glycerol. So you can see this ACE2 activator protein binds at this place. Other proteins are also there, but this protein is also bound at this. This acts as a repressor of the system, and this can only be activated when there is a phosphorylation of this ACE2 by some other protein, and this is done in the presence of xylan, xylobiose, or even sulfurose. So all these are inducers of this xylanase gene expression, and here you can see a lot of gene expression. This repressor protein also detaches from here, and these activators are there. So in this way we see how these two xylanase genes levels, they interact with different transcription factors for their repression or activation of the genes. Now I discuss a very interesting gene expression in archaea, which are single-celled microorganisms.
They behave like bacteria in many cases, just like uh, prokaryotes, and because they don't have any cell nucleus on any other organelles. But we will see in some ways they also uh, behave like eukaryotic cells. So how this takes place? Archaea are basically classified as a distinct domain of life other than eukaryota and prokaryotes. So they are distinct domains due to their certain properties. And they exist in a broad range of habitats and major global ecosystems. They contain this archaea and may contribute up to 20% of the total biomass on the earth, which is quite significant. And they also exist in hot springs of Yellowstone National Park, US. If you know that there are hot spring geysers erupting, uh, some are erupting after every few minutes, other after every few hours, other after some after a few days, weeks, and so on. So these are hot geysers over there. So many of the archaea, they have been isolated from this extreme temperature environment. They are extremophiles, some of them, as they can resist very high temperatures, even more than 100 degrees Celsius, as found in geysers and black smokers. And also, on the other hand, very cold habitats, or they can also resist highly saline, acidic, or alkaline water. And there are certain mesophiles which exist in marshlands, sewage, and soil. And then there are methanogenic archaea, which are present in digestive tracts of animals such as ruminants, termites, and human beings. Archaea are usually harmless to other organisms, and non-pathogenic. They are salt lovers, halophilic, Archean species near San Quentin, Baja California, Mexico. Similarly, south end of San Francisco Bay, they exist in the high salt uh, levels. What are the similarities with bacteria? Many common characteristics. Both lack internal membrane structures. They use plasmids to pass information. Both have ability to live in very extreme and hostile environments. And ribosomes of archaea is structurally similar to bacteria. Interestingly, they have many similarities with eukaryotes. DNA with histones, DNA replication is very similar. Transcription, they have RNA polymerase 2 type of transcription for messenger RNA. Glycosylation similar to eukaryotes. Transcription factors to turn on their genes similar to the eukaryotes. Similarly, transfer RNA contains introns. And sensitivity of ribosomes to chemical inhibitors is also just like eukaryotic cells. RNA polymerase, as I mentioned, for the transcription is also very much similar. And bacterial adoption similarities are also there. So this makes these archaea very interesting and also very good medium to study eukaryotic processes, because these archaea are simple ones just like bacteria, but on the other hand the many processes going on inside the archaea are taking place just like eukaryotes. So very good host to study many eukaryotic mechanisms. Look at the difference between archaeal, bacterial, and eukaryotic promoter. So bacterial promoter, they contain -10 and -35 region, and this is the regulatory sequence. But in case of eukaryotic promoters, Tata box is there, and archaeal promoters also have Tata box similar to the eukaryotic promoters. So archaeal promoters are very much similar to eukaryotic promoters, as compared to this bacterial promoters. This shows that gene expression in case of archaea is similar to eukaryotic cells rather than the bacterial cells. So with this I conclude my lecture. Thank you very much.